8. Breeding in plants and animal, including marker associated selection. Animal breeding. In animal breeding, a population is a group of inbreeding individual, that is a breed or strain within a breed that is different in some aspect from other breed or strain. Typically, certain animals within a breed or design as purebred. The essential difference between a purebred and non-purebred animals is that the genealogy of purebred animals has been carefully recorded, usually in a herd book or stud book kept by some sanctioning association. The Purebred Association provides other services that are useful to their members to enhance their business. The selective breeding utilizes the natural variations in trait that exist among members of any population. The breeding progresses requires understanding the two sources of variation, genetics and environment. For some trait, there is an interactions of genetics and the environment. The difference in the animal's environment, such as amount of feed, care, and even the weather, may have an impact on their growth, reproduction, and productivity. Such variations in performance because of environment are not transmitted to the next generation. For most traits measured in domestic animals, the environment has a large impact on variations than genetic differences. For example, only about 30% of the variation in milk productions in dairy cattle can attribute to genetic effects. The remainder of variations is due to the environmental effect. Thus, environmental factors must be considered and controlled in selecting a breeding stock. Genetic variation is necessary in order to make progress in breeding successive generations. Each gene, which is the basic unit of heredity, occupies a specific location or locus on a chromosome. Two or more genes may be associated with a specific locus and therefore with a specific trait. The trait that can be observed directly, such as the size, color, shape, and so forth, make up an organism's phenotype. These genes are known as alleles. If paired alleles are the same, the organism is called a homozygous for that trait. If they are different, the organism is heterozygous. Typically, one of the allele will be expressed to the exclusion of the other alleles, in which case the two alleles are referred to as dominant and recessive respectively. However, sometimes neither dominant, in which the two alleles are called codominant. Although no complete knowledge of genetic makeup of any breed of livestock exists yet, the genetic variations can be used for improving stock. The researchers partition total genetic variations into additive, dominance, and epistatic types of gene action, which are defined in the following paragraph. The additive variation is easiest to use in breeding because it is common, and the effect of each allele at the locus just add to the effect of other allele at the same locus. The genetic gains made using additive genetic effects or permanent and accumulate from one generation to the next. Additive and dominance variations are caused by gene at one locus. The epistatic variation is caused by the joint effects of the gene at two or more loci. There has been little deliberate use of this type of genetic variation in breeding because of complex nature of identifying and controlling the relative relevant genes. Breeding objectives. The breeding objectives can be discussed in the terms of changing the genetic makeup of a population of animals, where population is defined as a recognized breed. The choice of breeding goals and design of an effective breeding program is usually not an easy task. Complicating the implications of breeding programs is the number of generations needed to reach the initial goal. Ultimately, the breeding goals are dictated by market demand. Sometimes the marketplace demands different products than that was defined as desirable in the original breeding objectives. When this happens, the breeders must adjust their program, which results in the less efficient selection than if the new breed goal has been used for beginning. For example, the consumers want leaner beef that is tender. Thus, their rancher has changed their cattle breeding programs to meet the new demand. These trends have gradually changes over the last few decades. For example, the Angus cattle are partially noted for the quality of beef produced. The use of ultrasound is now widespread in determining the fat and lean content of live animals, which will hasten the changing of carcass quality to meet customers' demand. Additional complication arises from simultaneous trying to improve multiple traits and the difficulty of determining what part of variations for such trait is under genetical control. In addition, some traits are genetically correlated, and this correlation may be positive or negative. That is, the trait may be complementary or antagonistic. The breeding methods depends on heritability and genetic correlation for desirable trait. The heritability is the portion of additive genetic variations to the total variation. The heritability is important because without genetic variations, there was no genetic change in the population. The genetic correlation occurs when a single gene affects two or more traits. The practical breeding consequences is that the selection for one trait will pull along any positive correlation trait, even though there is no deliberate selection for them. For example, selecting for increased milk productions also increase protein productions. Another example is selection for increased weight gain in broiler chicken, which also increases the fat content of the bird. When trait have a negative genetic correlation, it is difficult to select simultaneously for both trait. For example, as milk production is increased in dairy cows, the genetic selection it might more difficult for the high producing cows to conceive. Hybrid vigor or heterosis. The heterosis or hybrid vigor is the phenotypic superiority of a hybrid over either of its parent in one or more trait. The hybridization between unrelated strain generally leads to an increased vigor and fertility. Hybrid vigor. The term heterosis was first used by Shull in 1914, though heterosis was first studied by Cole Reuter in 1763, followed by Darwin in 1876. The heterosis is lost by inbreeding, self-fertilization. Heterosis is generally expressed as increase in size and yield, but it may be expressed in several ways, such as earlier or later flowering and maturity, greater adaptability, faster growth rate. Heterosis is observed both in cross and self-pollinated species. Genetic basis of heterosis. There are two genetic hypotheses to explain heterosis: dominance hypothesis and over dominant hypothesis. The dominant hypothesis stated that heterosis is the result of masking of a harmful effect of recessive alleles by the dominant alleles. According to over dominant hypothesis, the heterosis is the result of superiority of the heterozygotes. There is no conclusive evidence to support or to reject either of two hypotheses. Heterozygote at certain locus are superior to homozygote. It is generally believed that the heterosis is the result of dominance and epistatic interactions. Some genes may also show over dominance. Biochemical basis of heterosis. Biochemical basis of heterosis at the biological level. Heterosis may result in one or many ways. Some of the mechanisms known as far as reduced amount of single gene product, separate gene product by the two alleles, combined gene product, or the hybrid substances and effects in two different tissues. Heterosis is commercially exploited in the form of hybrid variety or synthetic variety. Heterosis have been extensively used in many crop species, vegetables, fruit trees, and in animals. A list of some examples of plants and animal species where heterosis is being commercially exploited. Method of selection: the type of selection or individual or mass selection, within and between family selection, the sibling selection, and progeny testing with many variations. Within family selection uses the best individual for each family for breeding. Between family selection uses the whole family for selection. The mass selection uses records of only the candidate for selection. The mass selection is most effective than heritable is high, and the trait is expressed early in the life, in which case all that is required is observation selection based on the phenotype. When mass selection is not appropriate, other methods of selection which makes use of relative or progeny and can be used singularly or in combination. The modern technologies allows use of all these types of selection at same time, which results in greater progeny, greater accuracy. Evaluation of animal: the method of ranking animal for breeding purposes have changed as statistical and genetical knowledge has increased. Along with the increase in breeding knowledge, advancement in computing has enabled breeders to quickly and easily process routine breeding evaluations, as well as to develop research needed to rank large population of animals. Evaluating and ranking as candidates for selection depends on equating their performance records to a statistical model. Accuracy of selection: in some cases, the accuracy of selection for a trait can be measured using a calibrated tool or a scale. Thus, measurement of such a trait can be replicated with high reliability. Alternatively, some traits are difficult to measure on an objective scale, in which case a well-designed subjective score method can be effective. Because close relatives share many genes, an example of a relative of candidate for breeding can improve the accuracy of selections. The more complete the genealogical record or pedigree, the more effective the selection process. A pedigree is most useful when the heritability of a trait is relatively low, especially for traits that are expressed later in life or in only one sex. The reproductive techniques can be used to increase the rate of genetic progress. These reproductive technologies reduces the generation interval and increases selection intensity by getting more than one male calf from superior female. Both superovulation and sex determinations are now commonly used procedure. The superovulation is also used when the breeder wants to increase the number of female calves from a evaluated cow. Progeny testing. Progeny testing is used extensively in the beef and dairy cattle industries to aid the evaluation and selecting the stock to be bred. The progeny testing is most useful when a high level of accuracy is needed for selecting sire to be used extensively in artificial insemination. Breeding system: crossbreeding. The crossbreeding involves the mating of animals from two breed. Normally, breed are chosen that have complementary traits that will enhance the offspring economic value. The other consideration in crossbreeding is heterosis or hybrid vigor, which is displaced when offspring performance exceeds the average performance of a parent breed. This is common phenomena in which increased size, growth rate, fertility, or displaced genetical dissemination similarity. Such increase generally do not increases in successive generations of a crossbreed stock. So pure breed lines must be retained for crossbreeding and for continual improvement in the parent breed. In general, there is more heterosis for trait with low heritability. The heterosis is thought to be associated and with the collective actions of many genes having same effect individually but large effect cumulatively. Inbreeding, mating animals that are relatively related causing inbreed. Inbreed is often described as narrowing the genetic base because the mating of related animals results in offspring that have more gene in common. Inbreed is used to concentrate a desirable trait. Inbreed is generally detrimental in domestic animals. Increased inbreeding is accompanied by reduced fertility, slower growth rate, greater susceptibility to disease, and higher mortality rate. As a result, producers cry to avoid mating related animals. This is not always possible, though, when long continued selection for the same trait is practiced within the same population, because the parents of future generation are the best candidate from the lost generation, and some inbreed tends to accumulate. The rate of inbreed can be reduced, but if inbreed depression becomes evident, some methods of introducing more diverse genes will be needed. The most common method is some form of crossbreeding. Inbreed depression. Inbreed depression may be defined as the reduction or loss in vigor and fertility as a result of inbreeding. The inbreeding is generally accompanied with the reduction in vigor and reproductive capacity, that is, fertility. Generally, there is a reduction in the size of various plant pods and in yield. In several species, the harmful recessive alleles appears after selfing, and the plants or line carried them usually do not survive. The effect of inbreeding are summarized below. For A, inbreeding results in appearance of lethal, sublethal, and subvital, reducing the survival and reproductive rate. Characteristics such as chlorophyll deficiency, albino, chlorino, etc., rootless seedling, defects in flower structures, etc. The plant carrying such characteristics are lost from the population. B. There is reduced in the vigor of population. The plant becomes weaker and shorter due to the general reduction in the size of various plant pods. C. Reproductive ability of a population decreases rapidly. D. The population rapidly separates into phenotypically distinct lines because of an increase in homozygosity, due to which there is a random fixation of various alleles in different lines. Hence, the line differs in their genotype and consequently in phenotype. This leads to an increase in the variance of population. E. Following inbreed, there is increase in homozygosity in each line. The consequence, the variations within a line decreases rapidly. The line which are almost homozygous due to continued inbreed and are maintained through close inbreed are known as inbreed line or inbreeds. Ultimately, after 7 to 8 generations of selfing, the line becomes almost uniform as they approaches complete homozygosity, 99% homozygosity. F. Inbreeding generally leads to loss in yield. Inbreed line that can survive and be maintained yield much less than the open pollinated variety from which they are derived. The degree of sibling depression varies from one species to another. Some species, example onion, cucurbite, etc., exhibit little or no inbreeding depression, while in some species such as carrot, alfalfa is very severe. On the other hand, the self-pollinated species are adapted to inbreeding and do not exhibit any inbreeding depression. The heterosis and inbreeding depressions are closely related phenomena. They may be regarded as the two opposite sides of same coin. The genetic theory that explains heterosis and explains inbreeding depressions. There are two main theories to explain the heterosis and consequently inbreeding depression. Line breeding. Line breeding is a system of breeding in which the degree of relationship is less intense than in inbreeding, and it is usually directed towards keeping the offspring related to some highly prized ancestor. The degree of relationship is not closer than half brother, half sister mating or cousin mating, etc. Line breeding is a mild form of inbreeding. Plant breeding. Plant breeding is the science of improving the heredity of plant for the benefit of humankind. The transmission from wild plant to a cultivated plant started early in the history of mankind. Several plants reproduce by strict self-fertilization, where pollination from a plant will fertilize reproductive cells or ovules of the same plant. Other plants only mainly allows cross pollinations, where the pollen from one plant can only fertilize a certain different plant. Asexual propagations, vegetative propagation can also occurs in plant. Example, runners from strawberry plants, which gives a new plant which is genetically identical in its parent plant. All these different change the way plant breeders work. Apomixis is the phenomenon that seeds are produced, but in an essentially asexual way, so that the parent and offspring belong to one clone, just as in the case of normal asexual propagation. Principles of plant breeding. The plant breeding is a science based on the physical of genetics and cytogenetics. It aims to at improving the genetic makeup of the crop plants. The following objectives of plant breeding are: 1. Higher yield. 2. Improve quality. 3. Disease and insect resistance. 4. Change in maturity duration. 5. Agronomic characteristics. 6. Photo insensitivity. 7. Synchronous maturity. 8. Non shattering characteristics. 9. Determinant growth. 10. Dormancy. 11. Moisture stress and salt tolerance. 12. Elimination of toxic substances. 13. Winter hardiness. Some well-known achievements are developed of semi-draft wheat and rice variety, mobilization of Indian canes, and production of hybrid and composite varieties of maize, jowar, and bajra. Breeding methods in self-pollinated crops. Various procedures like introduction, selection, hybridization, mutations are used for genetic improvement of crop plants. The choice of breeding methods mainly depends upon four main factors: 1. Mode of pollination. 2. Mode of reproduction. 3. Gene action. 4. Breeding objectives in crop species. Selection. In self-pollinated crops, the selection permits reproduction only in those plants that have the desirable characteristics. This is achieved by raising the next generation from seeds produced by the selected plants only. The seeds from the remaining plants are rejected. The selection is essentially based on the phenotype of plants. The two requirements of selections are: variations must be present in population, variation must be heritable. The purpose of selection is to isolate desirable plant types from populations. The selection is one of the two fundamental steps of any breeding program. The two basic steps are: progeny selection, evaluation of worth of plant on the basis of performance of their progenies is known as a progeny type. Louis D. Vilmorin developed the progeny type. Therefore, it is known as a Vilmorin isolation principle or Vilmorin principle. The progeny type serves two valuable functions. One, to determine the breeding behavior of plant, that is, whether it is homozygous or heterozygous. Two, to find out whether the characteristics for which the plant was selected is heritable, that is due to the genotype. The selection must be based on the phenotype. The relative contribution of a genotype and the environment to the phenotype phenotype of the selected plants can be determined through progeny test. If the phenotypic difference or due to the difference in the genotype, they will be present in the progeny as well as otherwise they will be absent in the progeny. The one pure line concept. A pure line is the progeny of single homozygous plant of self-pollinated species. All the plant in the pure line have the same genotype. The phenotypic differences within the pure line are due to the environment and have no genetic basis. Therefore, the variation within a pure line is not heritable. Pure line selection. A pure line is the progeny of a single homozygous self-pollinated plant. In pure line selection, many plants are selected from a self-pollinated crop and are harvested individually. Individual plant progenies from them are evaluated, and best progeny is released as a pure line variety. Therefore, pure line selection is also known as individual plant selection. A pure line variety is a variety obtained from a single homozygous plant of self-pollinated crop. Characteristics of pure line. All the plants within a pure line have the same genotype as the plant from which the pure line was derived. 2. The variation within a pure line is environment and not heritable. 3. The pure line becomes genetically variable with the time. The genetic variation is produced by mechanical mixture, natural hybridization of mutation. Merits of pure line selection. 1. The pure line selection achieves the best possible improvement over the original variety. This is because the variety is the best pure line present in the population. 2. The pure line varieties are extremely uniform since all the plants in the variety have the same phenotype. Such as uniform variety is more likely by farmer and the consumers than a less uniform variety developed through mass selection. 3. Due to its extreme uniformity, the variety is easily identified in seed certification program. Demerits of pure line selection. The varieties developed through pure line selection generally do not have wide adaptation and stability in production processed by the local varieties from which they are developed. 2. The procedure of a pure line selection requires more line space and more expensive yield trial than mass selection. 3. The upper limit on improvement is set by the genetic variation present in the original population. 4. The breeder must devote more time to pure line selection than mass selection. These leaves have less time for their breeding program. Mass selection. Many plants of similar phenotypes are selected and the seeds are mixed to constitute the new variety. The plants that are selected based on their appearance on or phenotype. Therefore, the selection is done for easily observable characteristics like plant height, ear type, grain color, grain size, disease resistance, tillering ability, logging resistance, shattering resistance, etc. Sometimes the yield of plants may be used a criterion of selection. If the population has a variation for grain characteristics like seed color and seed size, the selection may be done for them before the seed of selected plants are mixed. Generally, the plant selected is in mass selections are not subjected to progeny type test. In case of self-pollinated crop, mass selection has two major applications. 1. Improvement of daisy or local variety. 2. Purification of existing pure line variations. The mass selection has only a limited application for the improvement of self-pollinated crops. It is generally not used for the handling of segregating populations derived from hybridization. In cross-pollinated crops, the mass selection leads to avoid inbreed depression, loss in vigor and yield. The further, because of heterozygous nature of population, several cycles of mass selections may effectively be characterized. Mass selection procedure. First year 1 for variable population 200 to 2000 plants with a similar but desirable trials or selected. 2 the seeds from selected plants or composited. Second year 1 the composited seeds were planted in a preliminary yield trial along with a standard check. 2 the phenotype of the selected population is critically evaluated. 3 to 6 years the promising selections are evaluated in coordinated trials at several locations. 1 if outstanding release as a new variety. Seventh year seed multiplications for distributions. The mass selection P is self-pollinated crops coupled with progeny testing. This method is more useful than the mass selection without progeny testing. It is commonly used for maintaining the purity of pure line varieties. Eighth year seed multiplication for distribution. Merits. 1 the varieties developed through mass selections are likely to be more widely adapted than pure lines. It is generally accepted that a mixture of closely related pure lines is more suitable in performance over different environment than a single pure line. 2 extensive and prolonged yield trials are now necessary. This reduces the time and cost needed for developing a new variety. 3 the mass selection tries considerable genetic variations. 4 it is a less demanding method. The breeders can devote more time to other breeding programs. Demerits. The variety developed through mass selection shows the variety variations and are not as uniform as pure line varieties. Therefore, such varieties are generally less likely than pure line varieties. 2 the improvement through mass selection is generally less than the pure line selections. It is because at least some of the plant progenies which makes up the new varieties would be poorer than the best pure line that may be selected from among them. 3 in the absence of progeny test, it is not possible to determine the genotype of selected plants. As suggested by the Allard, the progeny test may be included in a mass selection program to overcome this defect. Hardy Weinberg law. It is a fundamental law of population genetics and provides the basis for studying the Mendelian populations. This law was independently developed by Hartley in 1908 in England, Weinberg 1909 in Germany. The Hartley-Weinberg law states that the gene and progeny frequency in a Mendelian population remains constant generation after generation. There is no selection, mutation, migration, or random drift. The frequency of a 3 genotype for a focus with two alleles, say capital A and small a, would be p square into capital A 2 pp into capital A small a and q square small a, where small p represents the frequency frequency capital A. The small q represents the frequency small a. The sum of small p and small q is 1. Therefore, p plus q is equal to 1. Such a population would be at equilibrium since the genetic frequency would be stable, that would not change from one generation to the next. The equilibrium is known as a Hardy-Weinberg equilibrium. Let us consider the single gene with the two allele, the capital A and small a, in a random mixing populations. There would be 4 genotype capital A small a. Suppose the population has a capital N individual, of which capital D individuals or capital A, capital H individuals or capital A small a, capital R individuals or small a, so that capital D plus H plus R is equal to N. That is therefore total number of alleles at this locus in the population would be 2N. Since everyone has 2 alleles at the single locus, therefore the total number of capital A alleles is equal to 2D plus HA. Capital A into capital A small a. The ratio 2D plus H 2N is therefore the frequency of capital A allele in the population, that is represented by small p. Similarly, the ratio 2R plus H into 2N in the frequency of allele small a is written as a Q. Therefore, capital P is equal to 2D plus H divided by 2N is equal to Q is equal to 2R plus H divided by 2N, that is equal to capital D plus 1 by 2 H divided by capital N is equal to R plus 1 by 2 H divided by N. Therefore, P plus Q is equal to D plus H plus R is equal to NB is equal to 1. Therefore, P is equal to 1 minus Q, or Q is equal to Q minus P. The value of P and Q are known as a gene frequency. The gene frequency is the population of allele or capital A or small a in the population. The genotype frequency, the zygotic frequency in the proportion of genotype capital A or capital A small a or small a small a. The factors affecting the equilibrium in population. The equilibrium is a random mating population is distributed by migration. It is the movement of individual into a population from a different population. The migration may introduce new alleles into a populations or may change the frequency of existing alleles. To mutation, it is a sudden heritable changes in a organism and is genetically due to structural changes in the gene. The mutation may produce a new allele does not present in population and may changes the frequency of existing allele. 3. Random drift. It is also called as genetic drift, is the random changes in the genetic frequency due to sampling error. 4. Inbreeding. Mating between the individual sharing common parent in their in the ancestry is known as inbreeding. It reduces the proportion of heterozygotes and increases the frequency of homozygotes. 5th, selection. A differential reproduction rate of various heterozygotes is known as selection. The selection allows the selected genotypes to reproduce while undesirable genotype is determined. Thus, the breeders can improve the various characteristics by selecting for desirable types. The selection is a random mating population is highly effective in increasing or decreasing the frequency of allele, but is unable to either fix or eliminate them. However, in combination with a system of inbreeding, the selection is highly efficient in the fixation and elimination of alleles. Most of the characteristics of economic importance are quantitative characteristics and are governed by many genes. Such characteristics shows a continuous distribution, selection of extreme phenotype, increase the frequency of desirable alleles in population. The breeders has two basic tools to change the genetic composition of population. 1. Selection, and 2. Mating system. 1. Random mating. Each female gamete is equally likely to write with any male gamete, and the rate of reproduction of each genotype is equal. That is, there is no selection in such situation. A. Gene frequencies remains constant. B. Variance for the characteristics is constant. C. The correlation between relatives or prepotency does not change. 2. Genetic associative mating. The mating in between individuals that are more likely closely related by the ancestor in random mating. This mating system is more commonly known as inbreeding. It increases homozygosity. It is helpful in development of inbreeds, both partial and incomplete. 3. Genetic associative mating. Such individuals are mated which are less closely related by ancestry, then they would be under random mating. Thus, in this system, totally unrelated individuals are mated. Examples of such a mating are intervarietal and interspecific crossing. This system would reduce the homozygosity and increase heterozygosity. 4. Phenotypic assortative mating, the mating between the individuals which are phenotypically more similar than would be expected under the random mating is called phenotypic assortative mating. This is useful in the isolation of extreme population. Fifth, the phenotypic disassortative mating. The mating between the phenotypically disassortative mating is referred to as a phenotypic disassortative mating. It is very helpful in making the population stable and making the population stable, that is maintaining variability. The suitable parents may be selected to remove their weakness. The progeny from such a mating would be more desirable than the parent. It is also useful when the desirable type is an intermediate one and the available parents has have the extreme phenotype. Breeding methods in cross-pollinated crops. The population of cross-pollinated crop species are highly heterogeneous as well as heterogeneous. The population of cross-pollinated crops species are highly heterozygous as well as heterogeneous. Their genetic makeup is such that they shows variable inbreed depressions. Consequently, the breeding methods for cross-pollinating crop aims at preventing inbreed. The breeding methods commonly used in cross-pollinated crops may be grouped into two broad categories. One, population improvement. Two, hybrid and synthetic varieties. In case of population improvement, the moss selections or its modifications are used to increase the frequency of desirable adults, thus improving the characteristics of population. In case of hybrid and the synthetic varieties, a various number of strains are crossed to produce a hybrid populations. The strains that are crossed are selected based on their combining ability. The population improvement methods may be grouped into two general classes, without progeny testing and two with progeny testing. Without progeny testing, the plants are selected based on their phenotype and the number of progeny tests is carried out. Example, mass selections. Where population improvement includes the progeny selections or year two row method and recurrent selections. Mass selections. In this, it is the oldest breeding scheme available for cross-pollinated crops. In mass selection, a few plants are selected based on their phenotype and the open pollinated seeds from them is bulked together to raise the next generation. The selected plants can open pollinated, that is the mate at that is to mate at a random selection of plant is based on their phenotype and no progeny test is conducted. The selection cycle may be repeated one or more times to increase the frequency of favorables. Such a selection scheme is generally known as a phenotypic recurrent selections. The care should be taken to select a sufficiently large number of plants in order to keep inbreed to a minimum. The efficiency of mass selection primarily depends upon the number of genes controlling the character, the gene frequency, and more heritable. 1. Several plants selected based on the phenotype. 2. Open pollinated seeds from the selected plants grow. Yield trial. 1. Bulk seed from the selected plant grow. 2. More selection may be repeated. Merits of mass selection. 1. The work of breeder is kept to be minimum since the selection are based on the phenotype of plants. 2. The selection cycle is very short, that is only one generation. 3. It is highly efficient in improving characteristics that are easily identified visually and have high heritability. Example, plant height, size of the year, date of maturity. Demerits. The selection of plant is based on the phenotype of individual plants. Superior phenotype is often a poor basis for identification of a superior genotype. The environment affects the quantitative characteristics. 2. The selected plants can be open pollinated, so the selected plants are pollinated by both superior and inferior plants present in the population. This reduces the effectiveness of selection. Modification of mass selections. The two defects of mass selections are: 1. Lack of control on pollen source. 2. Confusing effect of environment on the phenotype of individual plant. The effectiveness of mass selection. It has effectively improved the characteristics with high heritability in maize. Example, ear height, lodging resistance, ear type, adaptiveness, oil and protein content, the resistance to leaf blight, and days of flowering. Heterosis and inbreeding depressions. The cross-pollinated and reproducing asexual species are highly heterogeneous. These species usually shows a severe reduction in fertility and vigor due to the inbreeding. Conversely, hybridization between the unrelated strain generally leads to an increased vigor and fertility. Inbreeding. It is a mating between the individual related by descent or ancestry. When the individuals are closely related, examples in brother-sister mating or sib mating, the degree of inbreeding is high. The highest degree in breeding is achieved by selfing. The selfing reduces heterozygosity by a factor of 1/2 in each generation. The degree of inbreeding increases in the same proportions. Inbreeding depressions, if it may be defined as a reduction or loss in vigor and fertility as a result of inbreeding. Hybridization, the crossing of two plants or lines of dissimilar genotype, is known as hybridization. The seed as well as a progeny resulting from the hybridization are known as a hybrid or F1. The progeny of F1 obtained by selfing or intermediate of F1 plants and subsequent generation are termed as a segregating generation. The term cross is often used to denote the products of hybridization, that is the F1 as well as the segregating generation. Objectives. The chief objectives of hybridization is to create the genetic variations. When two genotypically different plants are crossed, the genes from both the plants are brought together is F1. The segregation and recombination produces many new gene combinations in F2 and later generation, that is a segregating generation. The aim of hybridization may be the transfer of one or few qualitative characteristics or use of F1 as a hybrid variety. The combination breeding. The main aim of combination breeding is the transfer of one or more characteristics into a single variety from other variety. The familiar examples of combination breeding is that the disease resistance. In combination breeding, one of the parent must have in an enough intensity the character under transfer, while the other parent is generally a popular variety. The transgressive breeding. The aim at improving yield or its contributing characteristics through transgressive segregation. The transgressive segregation is the production of a plant in an F2 generation that are superior to both the parent for one or more characteristics. Such as plants are produced by an accumulation of pulse or favorable genes from both the parents at a consequence of recombination. The parent parent involved in hybridization must combined well with each other and should be genetically diverse. That is quite different. Each parent is expected to contribute different plus genes, which when brought together by recombination gives rise to the transgressive segregant type of hybridization. One, intervariational hybridization. The parent involved in the hybridization belonging to the same species. They may be two strains, the varieties or race of same species. It is known as a intra-specific hybridization. In crop improvement programs, the varietal hybridization is the most commonly used. The intra-varietal cross may be simple or complex depending upon the number of parents involved. Simple cross, the two parents are crossed to produce the F1. The F1 is selfed to produce F2. Complex cross, more than two parents are crossed to produce hybrid, which is then used to produce F2. Such a cross is known as a convergent cross because this crossing program aims at converging, that is bringing together genes from several parents into a single hybrid. In breeding of highly improved self-pollinated crops like wheat and rice, the complex cross are common practice today. As crop improvement progresses, the crop varieties would accumulate more and more favorable genes. To distant hybridization, it includes cross between different species of same genus or different genera. When two species of same genus are crossed, it is known as a intraspecific hybridization. Both when they belong to different genera, it is termed as intra-generic hybridization. Generally, the objectives of such cross is to transfer one or few simply inherited characteristics like a disease resistance to a crop species. Procedure of hybridization. There are 7 steps involved in hybridization. 1. Choice of parents. The choice of parents mainly depends upon the objectives of a breeding program. In addition to other program, the increased yield are always an objectives of the breeder. Therefore, the last one of the parent involved in a cross should be well adapted and proven varieties in the area for which the new varieties in beginning development. The other varieties should be having characteristics that are absent in this variety. Some parents produce superior F1S and F2S, while other do not. This property of parent is known as a combining ability. The combining ability of parent may serve as a useful guide in the selection of parents for hybridization program. Thus, the choice of parents is the basic steps in hybridization program, and often more than anything else they determine its success or failure. Evaluation of parents. If the performance of parents in this area where breeding is to be done is not known, it should be determined particularly for the characteristics they are expected to contribute for the disease resistance. The new strains should be checked for mechanical mixture and for heterozygosity. If it is suspected to be heterozygosity, it may necessary to self-pollinate a parent for one or more generation. 3. Emasculation. The removal of stamens or anthers or killing the pollen grains of a flower without affecting in any way the female reproductive organs is known as emasculation. The purpose of emasculation is to prevent the self-fertilization in the flowers of female parent. In dioecious plant, the male plant are removed, while in monoecious species, the male flower are removed to prevent self pollination. 4. bagging. Immediately after emasculation, the flower or inflorescence or enclosed in suitable bags of appreciate size to prevent random cross pollination. In cross pollinated plants like maize, the male flowers are also bagged to maintain the purity of pollen grains for pollination. Butter paper bags are the most commonly used. Fifth, tagging. The emasculated flower or tagged just after bagging. The tags are attached to the flowers or the inflorescence with the help of thread. The following information is recorded on the tag with the carbon pencil. A, date of emasculation. B, date of pollination. And C, date of female and male parent. 6, pollination, the two important operation that determines the amount of seed set in hybridization or emasculation and pollination. The in case of pollination, the mature, fertile and viable pollen should be placed on a receptive stigma to bring about fertilization. It is advisable that the fresh pollen from a mature anther should be used for pollination. Seventh, harvesting and storing the F1 seed. The crossed head or pods should be harvested and threshed. The seeds should be dried and properly stored to protect them from storage pest. The seeds from each cross should be kept separately and preferably kept with the labels. Introgression, the transfer of a few genes from one species into the full diploid chromosomes complement of another species. Polyploid in plant breeding. The somatic chromosomes numbers of any species, whether diploid or polyploid, is designated as a 2n, and the chromosome number of gametes is denoted as a small n. An individual crossing the gametic chromosome number small n is known as a haploid. A monoploid, on the other hand, has the basic chromosome number x. In a diploid species, n is equal to x. One capital X constitutes a genome or a chromosome complement. Clonal selection. Some agricultural crops and many horticultural crops are asexually propagated. Some common asexual propagated crops are sugarcane, potato, sweet potato, colocasia, Dioscorea, gams, mentha, ginger, turmeric, banana, etc. Almost all the fruit trees. The segregation and recombination procedure, new gene combination due to which the progeny differs from their parents in genotype and phenotype. A sexual reproduction, on the other hand, produces a progeny identical to their parents in the genotype because the progeny is derived from vegetative cells through mitosis. It prevents the genotype of an individual indefinitely. Any genotype is preserved and maintained through asexual reproduction. Characteristics of asexual propagated crops. 1. A greater majority of them are perennials, example sugarcane fruit trees. 2. Many of them shows reduced flowering and seed set. 3. They are invariable cross-pollinated. 4. These crops are highly heterozygous and show severe inbreeding depression. 5. The vast majority of asexually propagated crops are either polyploid, example sugarcane, potato. 6. Many species are interspecific hybrids, example banana and sugarcane. And 7. This crop consists of many clones, that is progeny derived from a single plant through asexual reproduction. Clone. A clone is a group of plants produced from a single plant through asexual reproduction. Thus, the asexual propagation crop consists of many clones. These crops are also known as clonal crops. All the members of the clone have the same genotype as the parent plant. As a result, they are identical with each other in genotype. Consequently, the phenotypic difference within the clone do not have a genetic basis and are purely due to the environmental effects. Characteristics of a clone: 1. All the individual belongs to the single clone or identical in genotype. 2. The phenotype variations within a clone is due to the environment only. 3. The phenotype of the clone is due to the effect of genotype capital G, the environmental capital E, and the capital G into E interactions over the population means smaller. Therefore, phenotype capital P is equal to capital N plus capital G plus capital E plus GE. 4. Theoretically, clones are immortal. That is, a clone can be maintained indefinitely through asexual reproduction, but the clone usually uh, degenerate due to the viral or bacterial infections. 5. The clones are generally highly heterozygous and show severe loss in vigor due to the inbreed genetic variations within the clones. It may arise due to the somatic mutations, mechanical mixture, and occasional sexual reproductions. Clonal de- degeneration. Theoretically, the clone are immortal. The loss in vigor and productivity of clone with time is known as a clonal degeneration. The clonal degeneration may result from one mutation, two viral disease, three bacterial disease mutation. It is a recurrent process. It may becomes a problem over a long period of time. Viral disease, they are easily transmitted through vegetative propagation. The virus are perhaps responsible for more cases of clone degenerations than any other single cause. Bacterial disease, by the results of bacterial infection making clonal degenerations. Method of improving of asexual propagation crops: a single outstanding plant selected from a population forms the basis of new variety. The breeding behavior or genotype of plant is not important. Since there would be no further sexual reproduction, the outstanding plant may be selected from an old unimproved varieties and improved varieties that have become varieties or from a population produced by crossing two or more clones. The procedure of selection used for such crops is known as a clonal selection, since the selected plants are used to produce new clones. In the earliest stage of clonal selections, when selection is based on single plant or single plot, the emphasis is on the elimination of weak and undesirable plant or clone. The breeder cannot be reasonably hoped to identify the superior genotype at this stage. In the latter stage, when the replicate trials are the basis of selection, the emphasis is to identify and select the superior clones. Merits of clonal selections: 1. It is the only method of selection applicable to the clonal crops. It avoids inbreed depression and prevents gene combination present in the clone. 2. The clonal selection can be combined with the hybridization to generate the necessary variability for selection. 3. The selection scheme is useful in maintaining the purity of clone. First year, few to several hundred superior plants selected. Second year, the clones from the selected plants grown separately, desirable clones selected. Third year, the PYT with the quality studies resistance scored, outstanding clone selected. And fourth to seventh year, the multi-location trial standard checks, the best done identified for release. And eighth year, the seed multiplication for the best done. Demerits of clonal selections: this This method utilizes the natural variability already present in the population. It is not devised to generate variability. Two, sexual reproduction is necessary for the creations of variability through hybridization. Wide hybridization. The hybridization between individuals from different species belonging to the same genus or two different genera is termed as a distant or wide hybridization. When individual belonging to crossed belonging to the species forms two different genera, it is referred as a intrageneric hybridization. When individual from two distinct species of same genera or crossed, it is known as a interspecific hybridization. Breeding procedure for wide hybridization. 1. Back cross breeding. When interspecific crosses between the two species of varying ploidy level are made, invariably the hybrid are sterile. By chromosome doubling with the application of colchicine, amphidiploid can be produced. Such amphidiploid or fertile cultivated tobacco, Nicotiana tabacum 2n is equal to 24, which crossed to n. The glutinosa 2n is equal to 12 produces a sterile F1 by the chromosome doubling and amphidiploid n. Digluta 2n is equal to 36 was produced. This was reasonably hybridizing with n, capital N. The tabacum by repeated backcrossing a mosaic resistant line with 2n is equal to 24 was developed. 2. Amphidiploidy. The man-made cereal triticale is an intragenic allopolyploid combining triticum aestivum with 2n is equal to 24 and scale cereal rye 2n 14. Rhabdobrassica was synthesized by crossing Raphanus sativus radish 2n 18 and Brassica oleracea cabbage 2n 20. 3. Bridging species technique. When a direct cross between two species are difficult, a third species is used in such cross. The hexaploid wheat triticum aestivum 2n42 does not cross with the diploid species. When T. dicocoides 2n14 and amphidiploid was produced, it crossed with T. aestivium 2n42. The Nicotina sylvestrin 2n42 is a bridging species to transfer the nematode resistant from N. rependa 2n48 to N. tacbacum 2n48. 4. Alien addition and alien substitution line. By crossing two unrelated species of different poly level and doubling the chromosomal number of sterile F1, the fertile amphidiploid are obtained. The amphidiploid is crossed back to the cultivated species repeatedly, twice or thrice, and then selfed. In the selfed progeny, the plants with one chromosome paired from the donor species in addition to the normal diploid chromosomes of this parent species may be present, and they are called alien additions line. In certain other plants, one chromosome pair of the donor species may substitute one chromosome pair of parent species when they are called alien substitution line. By adopting the above method, the mosaic resistance from the Nicotina glutinosa 2n is equal to 24, which are transferred to n tabacum 2n48 by alien addition 2n is equal to 48 plus 2, and alien substitution 2n is equal to 48 minus 2 plus 2. Mutation breeding. The mutation is a sudden heritable changes in a characteristics of an organism. The mutation produced by changes in the base sequence of a gene are known as a gene or point mutation. The term mutation were introduced by Hugo de Veris in 1900. The spontaneous mutation. The mutation occurs in natural population without any treatment by man at a low rate. These are known as a spontaneous mutation. The frequency of natural mutation is generally 1 is 10 lakhs. Induced mutation. The mutation may be artificially induced by treatment with certain physical or chemical agents. Such mutation is called induced mutation, and the agents used for producing them are termed as a mutagen. The utilization of induced mutation from crop improvement is known as mutation breeding. The induced mutation have a great advantages over the spontaneous one. They occurs at a relatively higher frequency, so that it is practical to work with them. Characteristics of mutations. One, mutations are generally recessive, but dominant mutations also occur. Two, the mutations are generally harmful to the organisms, but a small proportion, 0.1% of them, are beneficial. Three, the mutations are random, that is, they may occur in any gene. However, some genes show higher mutation rate than other. Four, the mutations are recurrent, that is, the same mutations may occur again and again. Fifth, the induced mutation commonly shows pleiotropy, often due to mutation in closely linked genes. Marker assisted selection. The marker assisted selection is a combined product of traditional genetics and molecular biology. The marker associated selection (MAS) allows for the selection of genes that control trait of interest. They combined with the traditional selection techniques. The marker associated selection has become a valuable tool in selecting the organisms for trait of interest, such as color, meat quality, or disease resistance. Molecular marker. Until recently, the researchers relied on information about how animals, plants, and their relative perform to make observation about the gene they possesses. Today, researchers can use a molecular marker to find genes of interest that control how plants and animals perform. Some molecular markers are pieces of DNA that have no known functions or impact on animal and plant performance. Other markers may involve the gene of interest itself. Link markers. One type of molecular marker is known as a linked marker. Using the well-designed experiments, the scientists may find the molecular markers that are located very close to major genes of interest. The molecular marker is said to be linked to that gene. The linked marker are only near the gene of interest on the chromosomes and are not part of DNA of gene of interest. Direct marker. A second kind of molecular marker is one that is part of gene of interest. The direct marker are easier to work with after they are found, but they often more difficult to find than linked marker. Three common technologies used as molecular marker are restriction fragment linked polymorphism, simple sequence repeats, and single nucleotide polymorphism. Restriction fragment linked polymorphism, RFLPS. The restriction fragment linked polymorphism, RFLPS, were the first molecular markers used to diagnose the genetic variability in organisms. The RFLP uses restriction enzymes to digest, to cut the DNA molecules and identify the regions linked to a trait. The number of DNA fragments generated by the restriction enzyme digest can be in the millions, with many being several thousand nucleotide long. This makes it difficult to determine the specific DNA fragments that are associated with the trait of interest on the electrophoresis gel. To help visualize the specific DNA fragments, a technique called southern blotting was developed. Simple sequence repeats or microsatellites. The simple sequence repeat SSRS, also called as a microsatellite, or repeated units of 2 to 6 nucleotide that occurs throughout an organism genome. This sequence AT is one example of a microsatellite. The sequence GAT is another example. The SSR, the simple sequence repeats, are useful as a molecular marker because they are highly polymorphic and have many forms. The single sequence repeats have been used successful as a marker in a wide range of analysis, particularly those involving disease diagnosis and forensics. Single nucleotide polymorphism. On average, the single nucleotide polymorphism will occurs in any organism DNA more than 1% of time, because only about 3% to 5% of them organism DNA codes for protein. The most single nucleotide polymorphism are found outside the region of genome of genes of interest. The single nucleotide polymorphism found in a gene of interest or the rest or the interest to research, because they are directly associated with the desired trait. Because of the recent advantages in technology, the single nucleotide polymorphism are playing a greater role in selection and diagnosis of a genetic trait. Advantages of molecular marker. The advantages of molecular marker for researchers is that they can test for a particular trait as early as the embryo stage in the animals or in seed of plant before they are planted. There is no longer a need for the organism to develop to stage at which the trait can be observed. A wait that is some case can take many years. Question: the plant breeding is a technique of improving uh, or agricultural crops, fodder crops, and fruit varieties, all the above. Answer: the plant breeding may be defined as the branch which deals with improving the heredity of crops and the production of new crop varieties which are superior to earlier over in all aspects.